faithfully conveying value and interpreting great books from around the world.
Hello, this is Sally's Book Talk.
Today we're discussing the book The Brain That Changes Itself, subtitled Brain Plasticity: Creating a Life Without Limits. Before we begin, let me ask you, do you believe someone born blind could ever see the world? Can someone born with only half a brain live normally? Could your memories be artificially erased? Surprisingly, the answer to all these sci-fi sounding questions is yes. That's the miracle of brain plasticity. Through rigorous scientific experiments, this book details how neuroplasticity works and its crucial roles in learning, therapy, rehabilitation, and technology. It helps you understand and unlock your brain's potential for a limitless life. The author, Dr. Norman Borage, is a renowned American psychiatrist and researcher at Columbia University's Psychoanalytic Center. Beyond his medical work, he's also a best-selling author, critic, and poet. And besides this book, his notable works include The Brain's Way of Healing. I'll break down the book's content into two parts for you. First, what is brain plasticity? Second, how can we harness brain plasticity? First, let's explore part one. What exactly is brain plasticity? Consider this real case to understand neuroplasticity more concretely. In 1959, a 65-year-old Spanish man suffered a stroke that paralyzed his right side. Doctors said he'd never recover, but his son created unconventional rehabilitation exercises. Inspired by infant development, he had his father practice crawling, then walking, coin tossing, dishwashing, and typing. Astonishingly, within a year, the man nearly fully recovered. He resumed work, traveled, mountain climbed, and regained all functions until dying from heart disease 7 years later. After his death, researchers performed an autopsy. Shockingly, the autopsy revealed that 97% of the nerves controlling the right side of the old man's body movement had died, causing his paralysis. Back then, the medical community widely believed that damaged brain function couldn't be recovered. Yet through exercise, the old man regained all abilities before his death. That's the miracle of brain plasticity. His brain must have undergone some changes. To understand how it changes, we first need to know how the brain is structured. The human nervous system splits into the central and peripheral systems. The central system includes the brain and spinal cord, acting as the body's command center, while nerves transmitting info belong to the peripheral system. This book focuses on the brain within the central system, where nerve cells, called neurons, play the key role. Neurons have three main parts: the cell body for storing genetic material, dendrites for receiving info, and the axon for sending it out. They transmit info as electrical currents, or signals, passing between neurons to create neural activity.
However, when adjacent neurons pass signals, the axon doesn't actually touch the other neuron's dendrites. But
There's a gap called the synapse, where neurotransmitters help the signal cross to excite or inhibit the next neuron. And so, stats show the human cerebral cortex has about 30 billion neurons, capable of a quadrillion synaptic connections. Clearly, synapses far outnumber neurons, so info transfer isn't one-to-one. One neuron gets signals from many and sends to many, forming a network, the neural circuit. Thus, among finite neurons, info paths can shift in many ways, letting the brain achieve a all-come-to-aid when one is in trouble effect. External factors like learning and training can alter synapses by adding or reducing connections, changing neural circuits. This is what we call brain plasticity. For example, the old man who recovered after a stroke. When the neurons controlling movement in his brain died, the connected synapses vanished, preventing movement commands from reaching muscles, causing paralysis. But through training, neurons in his brain that originally handled other tasks took over for the dead neurons. New synaptic connections formed between motor system neurons, allowing movement signals to be transmitted, restoring function. In other words, neurons in the brain are versatile. They're not stuck with fixed jobs, but can switch roles. Some listeners might have doubts here because it's often said that each brain region corresponds to one function. Others think each brain hemisphere has specialties, like the right brain handling spatial and abstract abilities, and the left excelling at logical reasoning. But is brain specialization really fixed from birth? Well, actually, no. Let's take another example. Michelle, born with only half a brain, yet living well at 29. His right brain took over functions for both hemispheres, operating the entire body. He has extraordinary calculation and memory skills, quickly recalling what day of the week a past date was. But his spatial and abstract reasoning is poor, showing that brain regions aren't limited to one specific sense. During development, the brain selects the most capable neuron groups for tasks, like the able do more work. For instance, the occipital lobe processes visual signals efficiently, making it the visual center. Over time, these neural circuits become relatively fixed but not unchangeable. With only half a brain, Michelle's neuron division of labor differs from normal, lacking half the resources. His existing neurons had to explore new pathways by forming unique synaptic connections and different neural circuits. And, for example, although his right brain is intact, he's not skilled in spatial or abstract areas. This is because during development, memory skills were more crucial for his survival. So he first developed memory skills, allowing memory functions that should be in the left frontal lobe to occupy the right frontal lobe instead. Thus, abstract thinking abilities like logical reasoning never had a chance to develop. This is like skiing down a mountain. Initially, we take different paths each time, but eventually a main trail forms. We always take that main trail down. If the main trail gets blocked, we can still find alternative paths. All right, we've seen how brain plasticity works through adding or removing synaptic connections to change neural circuits. So how can this brain feature transform our lives? Now, let's explore part 2, how to harness neuroplasticity through changing neural circuits. Specifically, there are 3 approaches: forming new neural circuits, correcting faulty ones, and sensory reassignment. I'll explain each approach in detail. First, the brain can achieve plasticity by forming new neural circuits. How exactly do we form new neural circuits? One method is creating new synaptic connections between neurons to establish new circuits. The best approach is through learning and training. Consider this experiment. Scientists recruited blind participants to learn Braille, attending classes Monday through Friday with weekends off. Using non-invasive methods, scientists mapped participants' brain activity. Brain mapping divides the brain into functional regions, like a geographical map. Neurosurgeon Wilder Penfield pioneered this concept in the 1930s while treating epilepsy and brain tumor patients. He discovered adjacent body parts have neighboring brain map regions, like thumb control areas next to index finger zones. After Braille training, scientists found learners' brain maps expanded significantly by Fridays during the first 6 months, but shrank back by Mondays.
However, after 6 months, Monday maps gradually grew until plateauing around month 10. After 2 months of rest, scientists measured participants' Monday brain maps again and found them nearly unchanged from before the break.
Researchers believe that during the first 6 months of Braille learning, rapid Monday to Friday map changes occurred because intensive training utilized existing synaptic connections, strengthening established neural circuits. After 6 months, the gradual expansion of Monday maps indicated entirely new synaptic connections forming between neurons, creating fresh neural pathways. Thus, the process was slow but remarkably persistent, remaining intact even after 2 months without practice. This parallels how we prepare for exams. When cramming last minute, we're using old neural circuits. We quickly forget after exams. Only consistent training creates new connections for lasting memory and improves neuronal efficiency.
For example, scientists trained monkeys to touch spinning disks with fingertips for 10 seconds, requiring precise pressure. Too much force would halt rotation. After thousands of repetitions, brain maps for their fingertips expanded significantly.
As monkeys mastered the task, fewer neurons were activated for the same action, showing increased neural efficiency. This frees up brain capacity for other tasks and new circuit formation. Beyond adding synapses, another approach is increasing neuron count itself. Traditional views held neurons couldn't regenerate, making quantity increases impossible. But recent research revealed neural stem cells in the hippocampus, critical for memory, that can create new neurons. These stem cells both differentiate into new neurons and self-replicate. So how do we increase neural stem cells? Two methods. First, exercise. Scientists tested mice running on wheels. After 45 days, their hippocampi were 15% larger than non-runners, with 15% more neurons. Thus, exercise promotes neural stem cell growth, increasing neuron count. Besides exercise, the second way to increase neural stem cells is learning. Studies show learning extends neural stem cell lifespan, boosting neuron count. The brain fears staying stagnant in unchanged environments. This accelerates its atrophy. Thus, the brain operates on use it or lose it. To keep it optimal, we must constantly learn new things instead of repeating mastered skills. This creates more neural circuits. When aging occurs, we'll have reserved circuits to rely on, another benefit of lifelong learning. So, how can we leverage the brain's ability to form new neural circuits? For instance, stroke treatment. As mentioned, forcing paralyzed limbs to move forms new neural pathways. Rewiring brain maps to restore limb function.
Note: focused intensive training yields best results.
Beyond this, we can also treat autism by forming new neural circuits. Autistic children often have extreme sound or touch sensitivity. Overwhelming sensory input makes coping difficult. Thus, they retreat inward. While genetics play a role, neuroscientist Merzenich believes environment greatly impacts development. From birth to early childhood, humans or animals have a critical period, peak sensitivity for skill development, highly influenced by environment. At birth, our brains resemble rough sketches. During the critical period, exposure to external information makes this sketch branch out and refine. Brain-derived neurotrophic factor, BDNF, crucially regulates this period. BDNF activates the basal ganglia, enabling easier neural connections and brain map changes, meaning critical period children learn effortlessly. When the critical period ends, BDNF secretion triggers switches that deactivate the basal ganglia, reducing new connections. So, autism may stem from premature critical period closure. At that stage, brain maps remain undifferentiated, neural circuits incomplete and chaotic. So even mild stimulation can trigger widespread neural activation, overwhelming the sensory system. Take a normally differentiated brain.
In its map, hearing the note do activates specific neurons, while re activates different ones.
But an undifferentiated brain activates the entire cortex with any sound. What causes premature critical period closure? Scientists suspect noise exposure might be responsible. Each sound excites auditory cortex neurons. Collective firing produces excess BDNF, triggering early critical period shutdown. Understanding autism's cause enables targeted solutions. Innovative therapy description. Merzenich developed software using repeated single tones like do to retrain autistic brains, activating specific neurons, then progressing to other tones, rewiring neural circuits to reduce symptoms, with proven success. Now, you might wonder, could we reopen adults' critical periods for easier learning? Technically, yes. Electrically stimulating or injecting substances into the basal ganglia could reactivate it. But such invasive methods risk brain damage, outweighing benefits. Beyond stroke or autism, forming new circuits treats learning disabilities, dyslexia, and brain injuries. The second plasticity method involves modifying faulty neural circuits. So, for faulty circuits, we weaken old connections while building new ones using the use it or lose it principle. How exactly? Consider OCD. Sufferers obsess over locked doors, clean hands, or orderly items, repeating actions tormentedly. But here's the insight. OCD stems from a locked brain. When we err, the brain follows 3 steps. First, the frontal cortex gives us the feeling of making a mistake. Second, the cingulate gyrus makes us anxious, prompting us to correct errors. Third, The caudate nucleus helps us automatically shift gears after correcting mistakes, but OCD patients show hyperactivity in all three regions.
It's like this neural circuit is stuck on, especially the jammed caudate nucleus, continuously signaling the frontal cortex about mistakes, causing repetitive behavior.
So how do we solve this? First, patients must detach from obsessions, observing themselves as impartial bystanders without engagement. For example, with OCD. When repeatedly checking locks, imagine watching yourself through a security camera from a detached perspective. Second, patients must recognize that persistent thoughts stem from faulty brain circuitry. The crucial final step, when obsessive thoughts arise, force attention toward meaningful activities like music, walks, sports, or social interactions to redirect focus. Beyond OCD, modifying faulty neural circuits also helps those with psychological trauma. The book describes Mr. L, whose mother died when he was 2. At 4, due to poverty and many siblings, his father sent him to a distant aunt's care. Within years, his world completely transformed. He adapted by enduring hardship, gritting through challenges as he matured. Yet, he remained emotionally distant, avoiding closeness. Through free association, therapists discovered his subconscious denial of his mother's death, who linked separation to the intense grief of her loss. Attachment to his mother made every separation feel catastrophic, ruining relationships and preventing intimacy. Therapy began by helping him recognize his issues originated from his mother's death. After her death, lacking familial support, he suppressed grief and built psychological barriers against memories. The therapist helped him break through this barrier, verbalizing his repressed grief about his mother's death. After the psychological barrier shattered, he gradually regained the feeling of having a mother. Well, he admitted missing her deeply and finally accepted her death. He released past sorrow and realized daily separations shouldn't trigger such intense grief. Gradually, those neurons stopped firing together, their connections severed, freeing Mr. L from separation anxiety.
Moreover, modifying faulty neural circuits effectively treats phantom limb pain.
What is phantom limb pain? After limb loss, 95% of amputees feel pain in the missing limb. That's phantom limb pain.
Illusory pain.
This pain is a neural illusion, like anorexia sufferers seeing themselves as overweight, despite being emaciated. The physical body and brain's body map aren't neurologically identical. Neuroscientist Ramachandran discovered phantom pain arises because amputated limbs can't execute commands, lacking feedback. The brain escalates signals, e.g., clench fist repeatedly without response. Eventually, imagining fingernails digging into palms creates pain, linking movement commands with pain neurons. He invented a mirror box, exploiting visual illusions to correct this. For lost left hands, patients place their right hand inside
Near's reflective.
left hand, tricking the brain into receiving movement feedback, stopping pain signals. Over time, faulty movement pain connections dissolve, relieving phantom pain.
Beyond OCD, phantom pain, and trauma, modifying neural circuits can change habits like unusual fetishes. The third plasticity method is sensory reassignment.
This means when a brain region can't receive signals from one sense, it can process information from other senses or devices, substituting for the blocked sense. In 1969, neuroscientist Paul Bach-Rita published a paper in Nature describing a tactile vision device for the congenitally blind. The device used tactile stimulators on the skin to help the blind see their surroundings. These stimulators vibrated to represent light intensity, brightness through touch.
Tactile signals activated the visual cortex, creating visual perception, allowing users to identify objects.
Later, skin stimulators were miniaturized into tongue electrodes, making the device portable. Thus, Bach-Rita concluded, "We see with our brains, not our eyes." He became the first to understand adult neuroplasticity and apply it clinically. Neuroplasticity similarly underpins Merzenich cochlear implants. Deaf users hear through microphones, converting sound to electrical signals sent directly to auditory cortex. Similarly, Balance-impaired individuals regain stability through tongue-based tactile feedback systems. Innovative Horizon enables advanced prosthetics. When touched, users feel sensations and perceive limb position, restoring touch and proprioception. All right, that concludes part 2. We've covered forming new neural circuits, modifying faulty ones, and sensory reassignment. These approaches harness neuroplasticity for trauma recovery, stroke, autism, OCD, phantom pain treatment, and restoring senses for the blind or deaf. But is neuroplasticity always beneficial?
Actually, no. It has negative impacts too.
For instance, shortened attention spans from binge watching short videos undermine deep reading. Fragmented media physically rewires our brains and cognition. Unlike deep reading and conversations, bite-sized content prioritizes speed and attention-grabbing formats. For instance, they use techniques like zooming, panning, overhead shots, and sudden noises. These abrupt changes trigger instinctive attention shifts, rewiring our neural circuits. Once accustomed to rapid shifts, slower pacing feels boring. Thus, deep reading becomes challenging, and neuroplasticity fuels hard-to-break addictions. Addiction includes not just drugs and internet, but porn, compulsive running, all behavioral dependencies. Addictive substances hyperactive dopamine, the neurotransmitter creating pleasure sensations. Dopamine also strengthens neural circuits associated with addictive behaviors. Merzenich experiments showed stimulating dopamine systems during specific sounds expanded auditory maps and sensitivity. With drugs, dopamine floods synapses, overstimulating nerves for intense euphoria. Long-term use desensitizes nerves to dopamine, requiring ever larger doses for the same high. To sustain dopamine euphoria, users crave drugs, cementing addiction. Simultaneously, dopamine reinforces the drug pleasure neural circuitry. Thus, during low moods, turning to drugs for relief becomes instinctive. Drugs also produce proteins that accumulate on neurons, hyperactivating dopamine systems, creating a vicious cycle of escalating addiction. These neural alterations persist even after quitting. Damage remains irreversible. This explains high relapse rates among recovered addicts. Recall how every experience etches itself into neural pathways. Childhood's peak plasticity is why early trauma shapes our entire lives. Studies confirm childhood trauma shrinks hippocampi, impairing long-term memory formation. Finally, the plasticity paradox is another negative effect caused by brain plasticity. It means that brain plasticity itself leads to the development of rigid, unchangeable behaviors. For example, why are bad habits so hard to break? It's because bad habits occupy specific areas of the brain map. At this point, good habits must compete for territory to squeeze in. Therefore, we should be extra vigilant against bad habits. Although change isn't impossible, it's very difficult. The discovery of brain plasticity has opened a new era, enabling us to tap into the brain's potential and create more miracles. With wise utilization, everyone can reshape their brain, transform themselves, and live a limitless life. All right, reshape the brain, reshape life. That concludes our interpretation of this book. Listening notes are below the audio. See you next time.